This talk is about the morphology and maturation of the Homo naledi ilium from the Lesedi Chamber. Homo naledi is known from abundant material from the Rising Star Cave system in South Africa. Fossils from the Dinaledi Chamber were first published in 2015, and these Dinaledi remains include nearly 2,000 commingled elements with over a dozen individuals represented throughout most of the skeleton. Dating to between 236 to 335,000 years ago, these remains reveal that a fairly primitive hominin survived until relatively recent times. Additional remains were subsequently discovered in other areas of the cave system, including the Lesedi Chamber. Most notable from Lesedi is a partial skeleton, Neo, which is anatomically consistent with Homo naledi from the Dinaledi Chamber. Note, however, that Neo has no pelvic remains aside from a fragment of the pubis. The pelvis of Homo naledi is represented by about 20 fragments, over 20 fragments from Dinaledi. These are quite fragmentary and none of them refit with one another, so it's unclear how many individuals are represented, although it is clear that there is a mix of mature and immature individuals. A few of the better preserved ilia, such as here in the center and in the top left, are complete enough to infer that Homo naledi had a wide and flaring ilium similar to the Australopiths. This primitive anatomy contrasts with a more derived and Homo-like ischium. Among the Lesedi fragments not associated with the neoskeleton is this immature ilium fragment UW102A-138, which I will just call the Lesedi ilium. Although it is much more complete than any of the Dinaledi remains, not much was said about it when it was published in 2017. We do know that it is immature because the acetabulum and anterior inferior iliac spine were unfused. Collected along with the Lesedi ilium were a number of fragments, including a large piece that we've subsequently identified as part of the iliac blade. This fragment, labeled B in these images, includes an abraded crest and superior iliac spine. So this refitting piece expands what is preserved in this already relatively complete specimen. Here's our reconstruction of the ilium with the original large fragment depicted in green and that refit fragment B in blue. Although there is some ambiguity as to the relative positions of these fragments, this arrangement here results in the most reasonable correspondence of surface contours between the two pieces. On the left side, we see the gluteal or external view, and on the right is the iliac or the internal view. We've highlighted areas where the cortical bone is abraded, such as the iliac spines, posterior margins of the auricular surface, and we'll note that the margins of the acetabulum epiphysis are also a bit abraded. Although much of the bone is missing, major anatomy is preserved, including the auricular surface of the sacroiliac joint. The gluteal view there on the left shows morphology that is consistent with the mature remains from Dinaledi. So the apparently compound acetabulocrystal and acetabulospinous buttresses is seen in one of the Dinaledi fragments, and the weak crystal tubercle is seen in another specimen. So here we have the Lesedi ilium compared with a human of comparable size, the most complete Dinaledi adult ilium, and an immature Australopithecus africanus from Makapansgat, South Africa. The images are to scale. So the smaller size and less developed epiphyses of Lesedi compared to the Australopith suggests that Lesedi is much younger. And compared with the Dinaledi adult, Lesedi implies a notable thickening of the acetabulocrystal buttress during growth. This all raises questions about the maturation stage and the morphological affinities of the Lesedi ilium. So we addressed the first question by comparing Lesedi and Dinaledi ilia with an ontogenetic series of humans, which we seriated by stage of maturation based on development of the acetabulum, anterior, inferior iliac spine, and the iliac crest. So we divided the humans into these 6 maturation stages, with 1, 2, and 3 roughly corresponding with infant, juvenile, and younger adolescent. Stage 4 representing older adolescent, and stages 5 to 6 being adult. Lesedi's acetabular surface is a bit too abraded to fully assess its maturation stage within 1 through 3. So we assessed a size measurement, the ilium height from the depth of the greater sciatic notch to the lateral margin of the crystal tubercle. Lesedi falls around the middle of the range of humans with their acetabulum unfused, whereas the only Dinaledi adult that preserves this measurement is similar to the smallest adult humans in our comparative sample. So to get an idea of Lesedi's maturation stage, we looked at its height proportional to the Dinaledi adult. And we compared this with immature and adult proportional heights, or immature divided by adult proportional heights among humans. We used resampling to randomly select an immature human in stages 1 through 4 and an adult human in stages 5 or 6 to get distributions of proportional size for each stage. The Lesedi proportional size is most likely to be sampled from the human stage 2 or juvenile distribution. This is important because the Lesedi ilium was found near other immature remains, including a mandible with teeth, and these are all developmentally consistent with one another, and so they may even be associated. We next wanted to compare the morphology of the Lesedi ilium with immature humans in Australopithecus africanus to test the hypothesis that Homo naledi retains a primitive ilium anatomy. And so we analyzed shape using three-dimensional geometric morphometrics. To describe shape, we made a template including 8 fixed anatomical landmarks in blue, 4 semi-landmark curves distributed across the iliac crest, the auricular margin, and the greater sciatic notch depicted in green. And then finally, 100 surface semi-landmarks on the iliac and gluteal surfaces depicted in yellow. We applied the template to 23 humans, 2 Australopithecus africanus, MLD7 and MLD25, all with the acetabulum unfused. And we used these as references for estimating the missing landmarks and missing morphology of Lesedi. So we've done this with this analysis with different reconstructions, different subsets of landmarks, and the results are generally the same regardless. So here we'll just show the results based on a Lesedi reconstructed with an Australopith model. In a principal components analysis in Procrustes form space, which incorporates both size and shape, Lesedi basically looks like a younger or smaller Australopith. On the right, the 3D model depicts the average shape of the immature humans, and compared with the humans, Lesedi and Australopithecus africanus have a thinner, flatter, and more anteriorly projecting iliac blade. But the superimposition of these ilium shapes shows one surprising result, which is that Lesedi in humans have a larger auricular surface of the sacroiliac joint compared with the Australopiths. So on the left, we've plotted the maximum length of the auricular surface against overall ilium size. And Lesedi falls right on the human regression, whereas the immature Australopithecus africanus are much smaller than expected for their size. So this is interesting because many hominins, including large specimens attributed to Homo erectus, have been described as having a small sacroiliac joint. And this unexpected similarity with humans in an otherwise small and primitive ilium raises questions about how Homo naledi transmitted its body weight during walking. So to summarize, the immature ilium from Lesedi is the most complete pelvic specimen of Homo naledi. Its overall size suggests it may have come from a juvenile of the small species, and it may even represent the same individual as other immature remains recovered in the Lesedi Chamber. This ilium also confirms the primitive morphology that was first identified in the mature ilia from the Dinaledi Chamber of the Rising Star Cave. However, this is the only Homo naledi that preserves the sacroiliac joint, or at least the auricular surface in its entirety. And this here suggests a more human-like anatomy compared with Australopiths or other members of the genus Homo. So, as with much of the Homo naledi skeleton overall, this individual ilium from Lesedi presents a mosaic of primitive and derived anatomy, whose meaning merits further investigation. We'd like to thank Vassar College, especially for support, including for undergraduate research, and we're grateful to the many people and institutions who allowed us access to skeletal and fossil remains. Thank you very much.